Good morning to all of you. So, last lecture, we have seen some basic part that was associated with the beta oxidation of fatty acids. Today, we'll try to learn certain errors that occurs because of beta oxidation inside the metabolism. So, we know that oxidation of palmitoyl coenzyme A, this is one of the important part in beta oxidation. Palmitoyl coenzyme A, 7-CoASH plus FADH2. This gives palmitoyl coenzyme A undergoes 7 cycles of beta oxidation to yield 8 acetyl coenzyme A. Acetyl coenzyme A enters citric acid cycle and can completely oxidize to CO2 and H2. So the complete oxidation of acetyl coenzyme A results in generation of ATP molecules. Now, energetics of beta oxidation. This is an important, very important slide because beta oxidation 7 cycles, FADH2, oxidation to ETC, it gives 14 molecules of ATP. 7 NADH gives 21. From 8 acetyl coenzyme A, oxidation to citric acid cycle, 96 ATP molecules have been generated. So total energy from one molecule of palmitoyl coenzyme is 131. And for all these purposes, for activation purpose of palmitoyl coenzyme A, some 2 ATPs have been utilized. Therefore, total net oxidation will be 121 molecules are being needed for the total oxidation of palmitoyl. Now, this is a part that is associated with why if the oxidation doesn't take place in a proper way, then what happens? Unexpected death of healthy infants usually overnight. During the deficiency of medium chain acyl coenzyme dehydrogenase. So enzyme specific for the conversion of acetyl coenzyme A is said to be very much important, playing an excellent role in it. Glucose is the principal source of energy, and soon after eating or feeding babies, after a few hours, the glucose level and its utilization decreases, and the rate of fatty acid oxidation must simultaneously increase to meet the energy needs. The sudden death in infant is due to blockade in beta oxidation caused by deficiency in medium chain of acyl coenzyme dehydrogenase. This disease is characterized by, now this is another disease, Jamaican vomiting sickness. This disease is characterized by severe hypoglycemia, vomiting, convulsions, coma, and death. It is caused by eating upripe fruit, which contains an unusual toxic amino acids, hypoglycemia. This inhibits the enzyme coenzyme A dehydrogenase, and their beta oxidation of fatty acid is blocked, leading to various complications. So there are certain molecules which act as blockers, or these block the beta oxidation of fatty acids, and because of this, certain complications would arise. Abnormalities in transportation of fatty acids into mitochondria and defects in oxidation leads to deficient energy production by oxidation of long chain fatty acids. So what are the features? Hypoketotic hypoglycemia, hyperammonemia, skeletal muscle weaknesses, and liver diseases. So these are abnormalities that would occur. Acyl carnitine, this is carnitine is said to be one of the important part which act as a carrier for the acyl molecules. Accumulates when the transfer or translocate locase is deficient. Dietary supplements of carnitine improve this conditions. Oxidation of odd chain fatty acids. Oxidation of odd chain fatty acid is similar to the even chain fatty acids. At the end, 3 carbon unit propenyl coenzyme is produced. Propenyl coenzyme, in that it was acyl coenzyme. This is propenyl coenzyme is converted into succinyl coenzyme. Succinyl coenzyme is an intermediate in TCA cycle. So propenyl coenzyme is gluconeogenic. Propenyl coenzyme is carboxylated to dimethyl malonyl coenzyme by biotin-dependent carboxylase. Biotin is B7 vitamin, and ATP is utilized in this step. Racemase acts on D-methyl coenzyme to give L-methyl coenzyme. This reaction is essential for entry of this compound into metabolic reaction of the body. Mutases that acts on to that particular thing, and is a vitamin B12 dependent enzyme. Therefore, it converts succinyl coenzyme into TCA cycle, converted to oxaloacetate, and then it is utilized for gluconeogenesis. See this. The whole mechanism of conversion of propenyl coenzyme A to succinyl coenzyme takes place. Ultimately, the succinyl coenzyme gets converted into the TCA cycle, and the energy which compounds are generated from TCA cycle. So, important errors of This propyl coenzyme carboxylase deficiency characterized by propionic acidemia, ketoacidosis and development abnormalities, malonyl maloic acid aciduria, two types of malonyl maloic acidemias, deficiency of vitamin B12, and this defect is due to the mutase or recibase kind of enzyme. There is there accumulation of methyl malonic acid in the body textiles. Correct? It is excreted by the urine. Symptoms: several metabolic acidosis, damage the central nervous system and growth retardation, fatal in earlier life. Some patient responds to treatment with pharmacological doses of B12. So if B12 has been taken, this disease can be recovered. Correct? So that's all from my side today. And we have learned the beta oxidation as well as odd number of oxidations through the whole process of beta oxidation of fatty